we go. Alright, so my project is called Pond Health, Pursuing Restoration and Sustenance of the Williston Pond through Studies of its Connected Aquatic Ecosystems. So here's a brief table of contents. So first I'm going to start with the essential question, then I'll move on to background on the abiotic and biotic components of the pond, a map of the collection locations, results, and solutions in future applications. So my essential question was, how does the health of the Williston Pond affect the lives of organisms in its ecosystem, and what can we do to solve these problems? So basically, I went around the town of East Hampton at 6 different locations taking samples of water, looking at the organisms that were in the water, and also testing the parameters of the water. So what makes up a healthy pond? So abundant populations of benthic macroinvertebrates, stable pH level, ammonia should not be present, nitrite should not be present, and actually nitrite was something that I tested for originally, but it showed up as 0 in every location, so from the further data after this it is not going to be referenced. And then phosphate levels should be limiting. So first off, benthic macroinvertebrates. So benthic macroinvertebrates are small aquatic animals and aquatic larval stages of insects, and some of the ones that are common in East Hampton that I found in some of the water samples were bioindicators, which assess and reflect the health of a specific ecosystem. So some that I found were biting midgefly larvae, which are actually tolerant of a range of water quality, planaria, which are generally sensitive to low water quality and pollution, copepods, which are vulnerable to water quality fluctuations and pollution, daphnia, which are highly sensitive to pollution and low water quality, so seeing those were a good sign. Gilled freshwater snails, which are sensitive to pollution and low quality water, and they require lots of oxygen. So at the top left, that is a gilled freshwater snail. On the top right, those are copepods. The bottom right is a planaria, and the bottom left is a daphnia. So this is a sample of a midgefly larvae from the Williston Pond under a microscope. Here is a freshwater gilled snail from the Williston Pond, and here's a daphnia. And this one actually has eggs, which is a good sign, and that's one of the most sensitive organisms. And those were in quite a large amount of abundance, which was good. I also observed the algae. So algae is photosynthetic and serves as food for many aquatic organisms. String algae was present in all the collections that I had. Blue-green algae, which are cyanotoxic bacteria, can be deadly if it is consumed. And they actually bloomed in the pond below Williston a few years ago, which was quite a big deal. And so algae blooms often occur when rain washes runoff from fertilizers, manure, and sewage into water bodies. So this is a sample of string algae from the Williston Pond. So you can see it's actually all these green filamental structures of string algae. The green circles are duckweed that were in the sample as well. So for the water parameters, first I looked at the pH, which is the potential of hydrogen in a liquid solution, and it specifies the acidity, the measure of hydrogen ions in a solution, and every 1 point increase and decrease in the scale indicates a 10 times increase or decrease in acidity. Fluctuations in pH can be detrimental to the health of aquatic organisms, and the pH levels are based around 7, less than 7 being acidic and more than 7 being basic. Too acidic invertebrates are not able to build their skeletal structures with calcium carbonate, which dissolves in acidic liquids. If it's too basic, too high, bacteria is unable to survive and break down the biological waste in the nitrogen cycle. So local levels around East Hampton should be around 7.5. The next parameter that I looked at was ammonia, which occurs naturally in ponds, and it is created when bacteria breaks down organic matter during the nitrogen cycle. And high levels of ammonia can actually burn the gills of aquatic organisms. And when both the water temperature and pH go up, so does the toxicity of ammonia. So levels should be 0. The last thing that I looked at was phosphate, which is vital for life, a necessary element for plant growth, and it is usually a limiting nutrient, where small amounts limit the plant growth to healthy levels, but too much may lead to rapid plant growth in algae blooms. So here are the 6 locations that I collected water from. One is the Williston Pond, 2 is the rubber thread pond below Williston Pond, 3 is down at the bottom left by South Street, 4 is next to the gym where it leads into there, 5 is Nashawanik, and 6 at the upper right corner is the lower mill pond behind like rifts in the mill buildings. So I collected 2 times at these 6 locations throughout East Hampton, and I looked at the results with a pond test kit. So the first collection was in about the end of September, and the next one was about a month later after a rainfall. And so that was something that I wanted to kind of look at and see how that affected my data. So pH typically lowers after a rainfall, which you can see in samples 1, 4, 5, and 6. It went down a little bit, and that's because sulfur dioxide and nitrogen oxides mix with water in the atmosphere, and when that water falls as rain, it's acidic, lowering the pH. In water body 2 and 3, though, there were rises in pH, which I am going to hypothesize is because excess nutrients were being flushed into those water bodies due to rainfall. The next thing I looked at was ammonia. So there were pretty low levels except for in water body 2, which probably came from the breakdown of biological waste from the chemicals, and it also is a low point. But there's also a lack of flow in that pond, especially this time of year, and that could lead to that high ammonia concentration. Also, one thing to note is that sample smelled very bad. Mr. Policia agrees with that statement, and it was like sulfur, very strong, so that was something that was interesting. Then next I looked at the phosphate levels, and quick reference, it should be 0.1, which would be about here, and it's not that. So that was very interesting to see. That was unexpected. And I'm assuming that is because of fertilizers around East Hampton being flushed into the water, and because they're all connected, those nutrients are being carried into each water body. And so at water body 5, though, there was a very rapid boost in phosphate, and I'm assuming that is because water body 2, which is rubber thread, leads into water body 5, and usually there's not really a steady flow between those two, but after rainfall, I'm assuming that picked up and carried a lot of nutrients out of that pond into Nashawanik. And those levels in water body 2 stayed the same, probably because of the nutrients being flushed into that pond from fertilizers. And so here is a healthy pond next to the health of the Williston pond. And so the pH levels are very good. About 7.5 should be where it is, and it was about 7.3, so that is good to see. Ammonia was very low concentration, a little bit above what it should be, but that is kind of expected with the fertilizers that are used around the edge of the Williston pond, and the organic compounds that are being broken down. And then also the phosphate levels were very high, also due to the fertilizers. So aquatic animals can tolerate high levels of phosphates, but the algae bloom that occurs after will be harmful, and there will be an instant lack of oxygen. Where algae can outgrow aquatic plants, there's also lack of light coming through to the bottom. And a spike in ammonia level would be toxic to aquatic organisms. And if the pH is too high, the skeletons of macroinvertebrates are unable to be formed, and if it's too low, the bacteria is unable to survive. So, I have a few sustainable solutions for the Williston pond. The first one being proper balance of shade and plants. Trees around the edge. Physical Plant just cut down a few trees by the gym this summer, which may kind of not be a good thing with more sunlight entering the pond, and also the dead leaves, when they fall, provide nutrients into the water, carbon, phosphorus, and nitrogen that can be used by the organisms there. And also the grass around the pond provides structure and cover for organisms, so if we let that grow up a little bit around the edge, that may be better, and also protects from unwanted chemicals and waste entering the pond. The next idea that I would have would be to reduce the use of fertilizer, which prevents excess nutrients in the water flush through rainfall and limits the phosphorus and nitrogen available for algae. So I think you would see those phosphorus levels kind of going down if we decrease the use of fertilizers. And lastly, a fountain. Fountains actually increase the oxygen in the water, keep the pH in balance, agitates the surface, increasing flow, regulating temperature, controlling the algae growth, enhances the fish habitat, and they also look very cool. So that could be the Williston fountain. Just an idea, but thank you very much. Any questions?
Once you leave it connected.
Yeah.
Are
Yes.
you going to share your findings with Mr. Hill and the physical plant?
I believe I will. Yes. I have some. Yeah, okay. I have some connections, so I'll see what I can do. I think that would look nice, like a blue light and like a green light, kind of like, you know, you walk across the bridge at nighttime. It look nice. Any other questions? Yeah, Miss Lucia.
So I noticed that our pond is fairly clear on top without a lot of green.
Yeah.
When I walk on the bike path, the other side
Yes.
is all green.
Yes.
What's going on there? They're connected. Right?
Yes, they are connected. So I did go over to that pond, and when I was collecting samples, that was all duckweed. And so I think with the extra ammonia, which was shown in my graph, is providing nutrients for that duckweed to really root into the water and really thrive. And as I know from a different project that I'm doing, duckweed really takes off quickly when provided the right amount of nutrients. So I think that's why.
So the flow in of the ammonia is below our pond?
Below our pond, yes. Yeah.
So on your project, you did a lot of research. What made you decide to go to a certain different ponds, or how did you be able to make time for that? Especially since I saw, and I think I knew one. I think that was maybe number 6.
Mm-hmm.
That seemed a little farther away from campus.
Yeah. So I chose this specific area in watershed to because it was something that I'm passionate about. Like, I like fishing, and I've been to all these different ponds, and I've noticed like things like the duckweed, and there's a lot of differences between each body of water. So I wanted to kind of dig further into that. And also, like that you were talking about, the sample location 6, I thought it would be interesting to see, because that's like very far away from the other locations that I tested. So I think that data would have been beneficial to see the differences there, even though they still are connected.
Yeah, thank you.
Yeah.
How long overall did the whole research and then writing this all up take?
So we had a trimester to go from like coming up with an idea to presenting basically today. So about a trimester, and I had about a month in between to collect data, analyze that data, and then prepare to collect the next round and then analyze that afterwards.
Thank you.
So about a trimester, yeah.
How did you know when you were collecting the water samples that the organisms would be in it?
So I tried to pick like the area of each pond that I collected to be right along the edge, and I wanted to make sure that was consistent. And so when I came back to each location, I picked the same spot. But the organisms that I found were kind of an addition to the main project, which was looking at the water parameters. But I think it was also important to see the variety and abundance of those organisms that lived in a small water sample. Yeah.
Can you share anything about the sample by South Street?
Yes. So one thing about that sample was that there were no benthic macroinvertebrates at all in that sample, which was interesting to see. But actually there's not a graph of that. Oops. I believe that is due to the fact that that is the first body of water that connects to the rest, and that was kind of like a swampy, marshy area. And so some of those organisms I don't think were present in the water body until later in like the streams that flow. I can go back to the map actually and show you. This is kind of the beginning of this whole system, and it's composed of mostly rainwater. So I think those organisms weren't in that specific water body until over here, where I did get a few. Yeah.
the thing about the question about time, and if you had another month, another trimester to work on this, what would you do next?
I think I would probably do another round of collection. I think more data is always beneficial to have. But also I would look at like taking it to the next step and seeing how I can, like, talking to physical plant, asking about fertilizers, maybe even petition to see if we can reduce that, or get a fountain.
Outside.
Yeah.
Fountain.
Yeah, Ms.
So
Lucia.
that was another question that I had. I'm in favor of reducing the fertilizers,
Mhm.
but I know this plant probably isn't
Mhm.
because they're green lawns. So
Yeah.
attractive. So do you have any suggestions of what we could do to keep green lawns but not have the runoff into the pond?
I think it's difficult, especially with like the slope of the shape of the land around the pond, and kind of like everything that has rainfall will flush into the pond. So I don't really know a good way to get green grass. But I think we could try without maybe, and see what happens. But I'm not sure. I'll look into that. Anything else? No.
Let's give Tucker one more round of applause.